Hey everybody, we are looking here at section 13.3, which is all about DNA replication. And really, replication is just a fancy scientific term to mean copying. So when we talk about DNA replication, what we're really looking at is how a cell copies its DNA. It makes those sister chromatids really as part of the cell cycle, which is something, we've been talking about since chapter 11, where we first introduced the cell cycle and mitosis. So we're going to kind of step back here with this section and go back to that original cell cycle conversation and add some additional detail to part of that process. Okay. So if you will, again, go ahead and write down the title here, section 13.3, DNA replication. And we are going to kind of go through some of the more specific details of this process here. So we're going to talk about several enzymes that are going to be responsible for the actions and things that we need to have happen as part of this event in the cell cycle. So one of those enzymes is called DNA polymerase, and we're going to talk about that here in a few slides. But essentially, if you look at that word, you can kind of pull apart maybe its meaning. So DNA, it has something to do with DNA. Polymerase, so we see that there's a word there, poly or even polymer, if you remember from back in chapter 2, where we talked about monomers and polymers of biomolecules. So DNA polymerase is responsible for actually making polymers of DNA, and we'll look at that here in a few minutes. And then we're going to talk just briefly at the end of the section about how eukaryotes and prokaryotes differ in terms of how we synthesize and copy our DNA. Okay, so when we talk about replication, like I mentioned, this is just another word for copying. So this is actually the process that we talked about back in chapter 11 when I was trying to describe to you what's happening as part of the S, or the synthesis stage of this cell cycle. So what we said at that time is that that's where we're going to make sister chromatids. We copy our DNA, right? We synthesize our DNA, which essentially just means we're making more DNA. And as part of the stage here, now we can say, okay, since we're talking about the molecular side of this inheritance process, now we're going to actually look at, all right, how does that process actually happen? How do we get a pair of sister chromatids at the end of synthesis where, when we start out at the start of synthesis after G1, you only have one copy of everything. So it sounds like it's probably complicated, and it truly can be, but we're going to keep it for our purposes as simple as possible, okay? But when we talk about the cell cycle, just as kind of a reminder of what we've already discussed before, all cells typically go through the cell cycle. It can differ in terms of what M represents. Remember, if it's a cell that's eventually going to go through mitosis, we're talking about somatic or body cells. If it's a cell that will eventually go through meiosis, we're looking at germ cells, or those found in the ovaries and the testes. But regardless of what M represents, all cells will complete interphase if they intend to divide in one of those two ways. So as part of interphase, again, we have the gap stages, G1 and G2, and then what separates them is S, and that's where we are for this. So when we talk about DNA synthesis and copying DNA, I want you to keep in mind we are looking at specifically the synthesis stage, the S stage of the cell cycle. So in other words, in humans, right, we start out synthesis with 46 total chromosomes. When we get to the end of this S phase right here, right before G2, we should have a total of 92 sister chromatids, right? So if we keep in mind how that DNA number changes, it changes as a result of what's happening here. So let's look at exactly how we get that duplicated DNA, right? How we actually go about copying that molecule. So when we talk about replication, which is this, copying of DNA, DNA really doesn't do anything except it stays put in the nucleus. Like, its job is to store information, right? But it's not going to actually kind of be active in this process. What's actually going to cause replication to occur are going to be a class of enzymes that play a role in this process. So enzymes, remember, are a type of protein. Their role and their job in a cell is to help lower the activation energy. Remember, we talked about the height of that hill when we looked at chemical reactions back in chapter 2. Enzymes help to decrease the height of the hill, thereby making it easier to do a chemical reaction. So the enzymes that we're going to look at here, they're going to decrease the height of the hill and make it easier for these to occur, simply because they're present and they bring the correct things together. Okay, without an enzyme present, this may happen on its own, but it would be much too slowly to happen as it should during synthesis. Okay, so we need the help of enzymes, and in total, you need to know about 4 total enzymes. All right. So when we think about replication, those enzymes are actually doing all the activity that we need, all the different steps and events. And those steps I've got listed here, I think I have a total of just 3 steps. So here's 1 and 2. So the first thing that has to happen when we talk about copying and making sisters is we've got to open up our DNA double helix. So remember, as part of interphase, DNA is not compacted. It's not condensed into the form of those X-shaped chromosomes that we see in mitosis and meiosis. DNA is loosely organized. It looks like tangled spaghetti in the nucleus. And we can see that here in this image. I'm just looking at a double helix, right? But when we talk about replication in eukaryotes like us, we're talking about a good number of chromosomes that would look like this, right? I mean, that's a lot of tangly mess stuff in the nucleus. So helicase enzymes are going to play a role in helping to kind of unwind those, but only so far as we can actually, start to create a new strand. We're not going to unwind the whole thing at one time, okay? But per chromosome, you can have multiple helicases acting on the same linear chromosome, because remember, our chromosomes are many, many nucleotides in length. So helicase enzymes come in. Their job is to unwind the double helix, and to do that, what they're going to actually need to do is break the hydrogen bonds between our base pairs that normally sit here as our rungs of our ladder, right? So like here you can see there's where I would have my hydrogen bonds, and also over here. And remember, what's beneficial about this is that hydrogen bonds are actually pretty weak, so it doesn't require a lot of energy on the part of helicase to actually break those apart. Notice we are not breaking any of the covalently bonded sugars and phosphates here in the backbone, right? That's not the job here. We want to maintain that sequence. And we can do that if we can keep those single strands together. All right. What we're trying to separate are the double strands, right, to provide space to create some new molecules there. So within the nucleus, then, we have free-floating nucleotides, meaning they're just hanging out, waiting to be used in the nucleus. And these free-floating nucleotides, as chromosomes unwind, and we have this thing that we call a replication bubble here form, where it's basically they're separated and there's some space in between those 2 strands. These free-floating nucleotides will recognize that, and they'll come in and start to bind with and be attracted to the bases on the template strands. So my template strands here are represented by the blue ribbon. In other words, those are my original strands. That's what we started with. And the orange is going to represent the new strands. So this orange strand is showing I've got all these little nucleotides, and you can see them here with the orange color, that are coming in, and they're binding with their appropriate interacting partners, right? So A will pair with T and G will pair with C, just like we saw according to Chargaff's rules and Watson and Crick's structure. And that will continue to happen. So they hydrogen bond and interact with those matching base pairs. And then we'll have another enzyme that comes along called DNA polymerase. And DNA polymerase's job is to help kind of form the actual new strand. And so this molecule doesn't need any help in forming hydrogen bonds. Those are just attractive forces anyway. But what DNA polymerase will do, and these kind of lighter colored ovals here are supposed to represent that enzyme, DNA polymerase. Its job is going to be to form covalent bonds between the sugars and phosphates that make up the nucleotides of the new strand. So even though these things are coming in here and they're interacting with our new base pairing partners, they're not going to fully seal and create a nice consistent strand without the help of DNA polymerase. So again, like I mentioned earlier, polymerase, you can think about it as if we're creating this DNA polymer, right? We're making 2 in this case, one right here, this upper orange strand, and then this one down here at the bottom, right? So you can imagine where I started with 1 DNA double helix, as this process continues in both directions through this replication bubble formation, right? We're eventually going to end up with 2 separate chromosomes. All right, and that's exactly what's going to happen. So DNA polymerase helps with the formation of those covalent bonds. And then we have a third enzyme here called ligase, and ligase I like to think of as the glue that kind of holds everything together. So sometimes there are breaks in the new molecules, and that's for different reasons. It can be the way that we're synthesizing can create little breaks. It can be also be that we've got maybe mistakes that have to be fixed through proofreading, and that sometimes happens also. Regardless of the cause, ligase is going to follow DNA polymerase, and it's not being shown here, but it would be kind of behind. And ligase's job is going to be to seal up any breaks and piece together things as they may need to be in the formation of these new strands. So again, 3 of the enzymes that you need to be familiar with: helicase unwinds, polymerase makes a new polymer, and then ligase is going to seal up the breaks. Okay. So 4 steps, 3 different enzymes. And then I've got one additional enzyme we're going to talk about right here. So when we talk about this process, one of the terms that scientists like to use is called semi-conservative. So we like to say DNA replication is a semi-conservative process, which basically means we're conserving the original strands. We're still conserving the original double helix or the original chromosome. However, it's semi-conservative because we're going to pair it up with something that's new. So even though we saved the blue strands, and here's the same kind of image, it's just now I've taken what was going here left to right. I've taken that and I've kind of flipped it, right? Turned it on its side, so now it's going to be going vertically. So when we look at this, right, my blue strand is my old, my orange strand is my new, and so anytime we create a new strand, we're going to continue to pair it with one of the originals. The originals don't go away. So they are conserved, it's just we say semi-conservative because we're pairing them with something that's new, okay? So again, we get 2 new molecules of DNA, we'll get 2 new chromosomes, and each of those original molecules of DNA that made up that first double helix is going to be paired with a new strand. The last little piece of this is that we want to make sure, especially in eukaryotes, because we have what we call linear chromosomes, meaning unlike bacteria, our chromosomes are not circular, right? They have a start and they have a stop at either end. What tends to happen through this process is that the machinery, the enzymes that catalyze all of these events that we just talked about, can't make it to the end of the chromosome. So what I mean by that is they have to have something to hold on to. And when they get further and further along towards that very, very end, they don't have anything to grab onto. And so what inevitably will happen is they start to kind of fail to synthesize and copy the entire strand. They kind of fall off, and over time, that ending piece is going to get lost. Well, there are these kind of protective elements at the end of all of our chromosomes, for eukaryotes again, called telomeres. And if you remember, telo means end. So a telomere is a repeating sequence of nucleotides. In other words, it's just A, maybe like that. But when we talk about those repeating sequences, they really serve no purpose other than to provide space for that DNA polymerase, that helicase, the ligase, to hold on to that and ensure we're not going to lose genes that are, just above where that telomere is found. So telomeres, their job is to protect the genes that are found towards the ends of chromosomes, and to help make sure that telomeres are maintained, we use an additional enzyme called telomerase, all right, which is Telomerase. So telomerase enzymes are what are going to be functioning to help maintain telomeres at the end of chromosomes. And again, that's important to protect the genetic information that we find in those places, okay? So stepping back for a second to this semi-conservative replication process. So again, the blue strand is representing our original strand, the orange is the new. And so again, you end up with eventually, as this process continues this direction towards the top and this direction going downwards, you'll end up with 2 separate distinct chromosomes that ideally have the exact same sequence, right? Because our DNA strands are complementary, like we've seen before, okay? Which means where you have an A on one side, you have a T on the other, right? That's the idea behind those base pairing rules. Now, if we consider prokaryotes, we have something slightly different that's going to happen. So remember, prokaryotes have a single circular chromosome here. Prokaryotes, because they only have one chromosome, they're going to do this process a little bit more efficiently and quickly than we see in eukaryotes, but it's going to look slightly different. Again, we don't have telomeres here because it's not a linear chromosome, right, with a start and a stop. This thing is just continuous. So we have what we call a replication fork at either end. We have enzymes that function just like we see in eukaryotes, but again, they're slightly different because they're found only in bacteria. But those enzymes are going to catalyze the formation of this new strand, right? So we have new DNA here represented as the red. The original, again, is blue. But the idea is that eventually you're going to end up with 2 distinct and separate circular chromosomes as part of this process, right? And for bacteria, this would be used in leading up to something like binary fission. Remember, bacteria do not actually go through a cell cycle like we talk about with eukaryotes. But if they're going to divide and create more bacterial cells, they still have to be able to copy their DNA, right? And so they use this process here. So again, these replication forks are just the positions where enzymes are actively creating new strands, okay? Or where helicases may be unwinding that chromosome. Eukaryotic DNA replication, in contrast to that, like I said, our chromosomes are generally much larger and much more complicated than what we see in prokaryotes. And for that reason, to make things more efficient, we have lots of enzymes that are active at the exact same time. So if we think about a linear chromosome, and here we've turned it again vertically, you can have multiple replication forks, even more so than what we saw on the last screen. When we talk about these replication forks, that essentially means you can have a helicase that's going this direction, helicase that's going this direction, helicase this way, and helicase this way. And as a result, if you've got hundreds of those on a single chromosome, well, you can pretty quickly replicate that whole thing. It's better than starting at one end and trying to get through all maybe 100,000 base pairs, right? That would take a whole lot longer. So in general, eukaryotes use lots of replication forks, lots of enzymes active at the exact same time to copy DNA, okay? All right, so the last thing I want to do before I finish this section is to look at how we actually pretend as if we're going to replicate a DNA sequence. So how we write a complementary sequence if I give you one. And so I'm going to do that here up on the board. So I'm going to pause this and come back in just a second. All right. All right, so looking at exactly how we would replicate DNA, if asked to do so, because I can tell you you will be asked to do so on a quiz or even on your test. So what we want to think about is, how do our base pairs relate to this? And when we think about the DNA double helix, we're talking about base pairing rules, right? That A is always going to pair with T, and that G will always pair with C. And if you remember from what we talked about previously, we know that there are actually two hydrogen bonds between A and T. And remember, there are actually 3 hydrogen bonds that will form between G and C. But these are our base pairing rules that Watson and Crick came up with based on Erwin Chargaff's information and data, right? His quantification of the amounts of all these things in different living things. So this is what we need to keep in mind to be able to replicate DNA. And truly, this is not a complicated process, but essentially, you're going to be kind of mimicking what would be happening in a cell if, DNA replication was happening, right? So for this, I'm always going to start with an original strand. So I'm going to write original DNA. And when we replicate anything, in terms of DNA, we have different ends of a chromosome. And I know in one of the assignments you're doing or will be doing, that is discussed. But essentially, we have a 5' end and we have what we call a 3' end. And we know that within DNA, these strands run anti-parallel. So what that means is that if one's going this way, the other's going this way. Base pairing rules are still going to be the same. It's just when I go to write my sequences, I'm going to make sure that I've got my 5' end labeled, which is 5 with it's like a little apostrophe, and then my 3' end labeled here. And so my new strand is going to be 3' on this side, 5' on this side, okay? If that confuses you, don't get hung up on that too much. Just focus on the pairing stuff, all right? So if we think about original DNA molecule, let's say I have, I don't know, a good number of base pairs here. Maybe I'll do 15, like that. Okay, and so if I have that as my original DNA molecule there in blue, if I'm going to assume that a cell is going to go through DNA replication, then the DNA that I'm going to write as being produced through that process is what we call complementary DNA. And it's not like it's DNA that's giving complements to something. It simply means that this is DNA that pairs with the original molecule. Okay, or if we think about the original molecule as being a double helix, this would have been the other half anyway, right? So as we've talked about before, A always pairs with T. So my complementary DNA here is going to start with a T. T here in DNA will pair with the A in the complementary strand. G pairs with C. G pairs with C. A pairs with T. C pairs with G. G pairs with C. A with T. 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 A, C, G. Now, the only thing I left off there is what I mentioned before, which is that I need to label the ends of this molecule. So let me scoot this over just a little bit so I'm not writing everything on top of each other here. So complementary DNA. All right, so my 3' end of this molecule is going to be on this side. My 5' end of this molecule will be on the other side. And so again, what that means is we're going in this direction here. We always say DNA goes 5 to 3, and then we're going to go 5 to 3 here. So this is where we come up with that term anti-parallel. Base pairing doesn't change when we say that, right? It would be the same even if I was going parallel to it. But when we talk about the synthesis of this essentially means if my 5' end of this original DNA is over here, when I go to synthesize and create my new strand, I've got to start on this end and come this direction. That's it. So those polymerases, those helicases, ligases, they only work 5 to 3. Okay, they don't go in reverse. All right. So this is how you would set that up. When you're given a sequence, same idea. All that you're doing is matching up those A's with T's and G's with C's. All right. So hopefully easy enough. If you've got any questions, as always, let me know. Otherwise, it's the end of 13.3 and the end of chapter 13. So we'll be testing on this soon, and we'll be reviewing up until then. All right. So I will see you later. Bye, all.